Welcome to the deep dive. I'm thrilled for the journey we're about to embark on today. Our mission is to take a deep dive into the very heart of, well, life's instruction manual. We're pulling insights from a foundational chapter of Raven Biology of Plants, the 8th edition, specifically the chemistry of heredity and gene expression.
Yeah, it's a really foundational chapter. And for you, our listener, we're going to try and demystify how genetic information is, stored, copied, and ultimately expressed. How it leads to the incredible diversity of life all around us. We'll pull out those key aha moments, so you'll hopefully walk away feeling well-informed without needing the textbook open right in front of you.
Exactly. Think of this as your essential shortcut, kind of, to understanding how genes fundamentally work. We'll explore the elegance of DNA, the machinery that copies and translates it, and the sophisticated ways life fine-tunes its genetic expression. Let's unpack this. Okay, so the structure of DNA, life's twisted ladder. Before the 1950s, scientists knew genes were on chromosomes, right?
Right.
But there was this huge debate. Was it the complex proteins or the seemingly simpler DNA carrying the blueprint? The how was a big mystery.
Yeah, and this is where James Watson and Francis Crick famously come in. They weren't, typical lab bench scientists for this project. They were more like brilliant theoretical puzzle solvers. They sifted through all the existing data, X-ray images, chemical data.
Building models, too, right? Physical models.
Exactly, trying to deduce DNA structure.
And what they pieced together was, well, revolutionary. They knew DNA was a big molecule, long, thin, made of these repeating units called nucleotides.
And each nucleotide, as you probably know, has 3 parts. A phosphate group, that sugar called deoxyribose, and then one of 4 nitrogenous bases, adenine, guanine, cytosine, and thymine, AGCT.
Right, AGCT.
And what's really crucial here, structurally, is that these bases fall into 2 groups. Adenine and guanine are purines, they're a bit bigger, double-ring structures. Cytosine and thymine are pyrimidines, smaller, single-ring.
And that size difference matters, doesn't it, for the pairing?
It's critical. A purine always pairs with a pyrimidine across the ladder. It keeps the width of the DNA molecule constant.
Okay, and key clues came from others, too. Linus Pauling had shown proteins could form a helix.
Right, suggesting DNA might do the same.
And then Rosalind Franklin and Maurice Wilkins, their X-ray diffraction work was huge. It confirmed DNA was helical, like a spiral.
Absolutely. And Erwin Chargaff added another vital piece. He analyzed DNA composition from different species, and he found that the amount of adenine, A, was always pretty much equal to thymine, T, and guanine, G, always roughly equal cytosine, C.
Chargaff's rules, that 1-to-1 ratio, it was a massive hint about the pairing.
Exactly. So putting it all together, Watson and Crick figured out DNA isn't just one helix, it's 2. An entwined double helix.
Like a ladder, but then twisted into a spiral staircase, keeping the steps, the rungs, flat.
Yeah, that's a good analogy. The 2 sides of the ladder, the backbones, are made of alternating sugar and phosphate groups. And the rungs, those are the paired bases. And the big discovery was adenine only pairs with thymine. Guanine only pairs with cytosine, A with T, G with C.
And they're held together by hydrogen bonds, right? Weak bonds, but lots of them.
Precisely. 2 hydrogen bonds between A and T and 3 between G and C. Stronger pairing for GC. These hold the rungs together right in the middle of the helix.
And this specific AT-GC pairing immediately explained Chargaff's rules.
Perfectly. Plus, they figured out each strand has a direction, a chemical directionality, a 5' end and a 3' end. And the 2 strands run in opposite directions. They're antiparallel, like lanes on a highway going opposite ways.
Okay, antiparallel. And maybe the most important thing about this structure, the 2 strands are complementary.
Yes, that's the key. If you know the sequence of bases on one strand, say ADGTC, you automatically know the other strand must be TCCAG.
Because A always pairs with T and G with C.
Right. And this complementarity isn't just neat, it's fundamental. It's how DNA can be copied accurately. It's how it can be repaired. It underpins everything.
Which leads us right into DNA replication, copying the master blueprint. The structure itself just screamed copying mechanism, didn't it? Watson and Crick even noted that, famously understated, in their paper.
They did. It was perhaps the understatement of the century. It has not escaped our notice. But yeah, the ability to make exact copies is essential for heredity. So when DNA replicates, the molecule locally unzips.
Those weak hydrogen bonds between the bases break.
Right, and the 2 strands separate, just like opening a zipper, but usually just in one region at a time.
And each of those separated strands then becomes a template, A guide?
Exactly, a template. Free nucleotides floating around in the cell nucleus come in and pair up with their complementary bases on the exposed templates. A finds a T, G finds a C.
So if the template has a T, only an A nucleotide can slot in opposite it on the new strand.
Correct. This ensures that each original strand builds a perfect copy of its former partner. The end result: two identical DNA double helices, where there was only one before.
And that's how genetic information gets duplicated, passed down through cell division. Amazing.
It really is. And this whole intricate process happens only once per cell generation during a specific window called the S phase of the cell cycle.
Now, you said the principle is simple, but the process is complex. Lots of enzymes.
Oh yeah, a whole crew of molecular machines. It always starts at specific DNA sequences called origins of replication. Think of them as starting lines.
And you need special proteins to get it going.
You do. Initiator proteins recognize the origins, and then enzymes called helicases come in to do the actual unzipping, breaking those hydrogen bonds, and unwinding the helix. Then other proteins, single-strand binding proteins, clamp onto the separated strands to keep them from snapping back together immediately.
Okay, so it's unzipped and held open. Then what builds the new strands?
That's the job of enzymes called DNA polymerases. They're the master builders.
Yeah.
But they have rules. They can only add nucleotides to the 3' end of a growing strand. So they only build in one direction.
Okay.
5' to 3'.
5' to 3'. Got it. And they need a primer, like a little starting block.
Exactly. They can't start from scratch. They need a short RNA primer laid down by another enzyme to add on to.
Interesting. RNA involved even in DNA replication.
Yeah. Now, here's a cool difference between simple cells, prokaryotes, and more complex ones, eukaryotes. Prokaryotes, like bacteria, usually have just one origin of replication on their circular chromosome.
Okay, one starting point.
But eukaryotes, like plants and animals, have these long linear chromosomes. Replication starting at just one point would take forever.
So they have many origins.
Lots of them. Along each chromosome, if you could see it happening, you'd see these multiple replication bubbles opening up simultaneously along the DNA.
Like little eyes opening up along the strand.
Kind of, yeah. And at each end of a bubble, where the DNA is actively unwinding and being copied, you have a Y-shaped structure. That's called a replication fork.
Okay.
And these forks move away from the origin in opposite directions. So replication is bidirectional. The bubbles expand and eventually merge.
Now, you said DNA polymerase only builds 5' to 3'. How does that work if the two template strands are antiparallel, one runs the other way?
Excellent question. This leads to a fascinating asymmetry at the replication fork. One new strand, called the leading strand, can be synthesized continuously. The polymerase just follows the fork as it unzips, building smoothly in the 5' to 3 direction.
Okay, smooth sailing for the leading strand.
But the other template strand, the lagging strand, runs in the opposite direction. The polymerase has to work on this one differently. It synthesizes the new strand discontinuously in short pieces. These are called Okazaki fragments.
Okazaki fragments. Okay.
Each little fragment is made 5' to 3', but sort of backwards relative to the overall direction the fork is moving. It waits for the fork to open up a stretch, then synthesizes a fragment back towards the origin.
So it's like backstitching.
That's a great way to put it, like backstitching along the lagging strand.
And then these fragments need to be joined up.
Exactly. Another enzyme, DNA ligase, comes along and seals the gaps, stitching the Okazaki fragments into a continuous strand.
Wow. And there are other enzymes, too, to stop tangles?
Yes, topoisomerases. As you unwind the helix, the DNA ahead of the fork can get supercoiled and tangled. Topoisomerases relieve this strain by cutting, swiveling, and rejoining the DNA strands. It's incredibly coordinated.
Okay, so DNA replication is sorted, but that just copies the information. How does the sequence, the A's, T's, G's, and C's, actually direct the building of proteins?
Right, that's the next big step, gene expression. And this is where DNA's sister molecule, RNA, really takes center stage.
RNA, ribonucleic acid.
Correct. Its role was suspected for a while because cells that make a lot of protein always have a lot of RNA. And crucially, while DNA mostly stays safe in the nucleus in eukaryotes, RNA is abundant out in the cytoplasm.
Where the protein synthesis happens.
Exactly. So RNA looked like the likely messenger.
How is RNA different from DNA again?
Two main chemical differences, and one structural one, usually. First, the sugar in RNA is ribose, not deoxyribose. Ribose has one extra oxygen atom.
Okay, ribose sugar.
Second, instead of the base thymine T, RNA uses uracil. Uracil still pairs with adenine, just like thymine does. So A pairs with U in RNA context.
Right, U instead of T.
And structurally, while DNA is typically that famous double helix, RNA is usually single-stranded. It can fold up into complex shapes, but it's fundamentally a single chain.
Single-stranded U instead of T, ribose sugar.
Got it. And there are different types of RNA.
Three main types are crucial for protein synthesis. First, messenger RNA, or mRNA. This is the molecule that actually carries the genetic code, the instructions from the DNA and the nucleus out to the protein-building machinery. The messenger. Makes sense. How is it made?
It's synthesized using a DNA strand as a template in a process called transcription. Similar base pairing rules apply: A with U, G with C. An enzyme called RNA polymerase does this job.
Okay, mRNA carries the code. What else?
Second, ribosomal RNA, or rRNA. This type of RNA doesn't carry code. Instead, it combines with proteins to form the actual ribosomes.
The ribosomes, the protein factories themselves.
Exactly. RNA is a structural and even catalytic component of the ribosome.
Wow, okay. And the third type?
Transfer RNA, or tRNA. These are the real adapter molecules. They act like a dictionary. Each tRNA molecule is designed to recognize a specific code word on the mRNA and bring the corresponding amino acid to the ribosome.
So tRNA reads the message and fetches the right building block.
Precisely. So you have this flow of information. DNA holds the master plan. Transcription creates an mRNA copy of a specific gene. Then translation uses RNA in ribosomes and tRNA to read the mRNA message and build a protein.
Replication, transcription, translation, the central dogma, basically.
That's the core flow, yes.
Okay, let's talk about that code. The genetic code, translating from nucleotides to amino acids.
Right. DNA and RNA use a language with just 4 letters: A, U, or T, G, C. But proteins are built from 20 different kinds of amino acids. So how do you get from 4 letters to 20 amino acids? That was the coding problem.
It's like cracking a cipher. If one nucleotide coded for one amino acid, you'd only have 4 options. Not enough.
Correct. If you use pairs of nucleotides like AU or GC, how many combinations would that give you?
4 times 4, 16. Still not enough for 20 amino acids.
Exactly. So the minimum number of nucleotides needed to specify one amino acid had to be 3. A 3-nucleotide sequence gives you 4 by 4 by 4.
64 possible combinations, plenty for 20 amino acids.
Right. So the hypothesis was that the genetic code is read in triplets of nucleotides. These 3 nucleotide units are called codons.
The triplet codon hypothesis makes sense mathematically, but how did they prove it and figure out which codon meant which amino acid?
That took some brilliant experimental work in the decade after Watson and Crick. Marshall Nirenberg and Heinrich Matthiei were key players. They synthesized artificial mRNA molecules with known sequences. For example, an mRNA made only of uracil.
Poly-U.
Right.
Okay.
They added this to a cell-free system that could make proteins along with all 20 amino acids, one of which was radioactively labeled. They found that the poly-U mRNA only produced a protein chain made entirely of the amino acid phenylalanine.
So UU must be the codon for phenylalanine, the first word cracked.
Exactly. That was the breakthrough. They then developed more sophisticated methods using different known sequences to decipher the rest of the code.
And what did they find? Out of the 64 possible codons.
61 of them specify particular amino acids.
What about the other 3?
The remaining 3 codons, UAA, UAG, and UGA, act as stop signals. They tell the ribosome to stop adding amino acids and terminate the protein chain.
Okay, 61 coding codons, 3 stop codons, but 61 is still way more than 20 amino acids.
Right. This means that most amino acids are specified by more than one codon. The genetic code is redundant or degenerate.
Like different spellings for the same word?
Kind of. For example, leucine is specified by 6 different codons. Phenylalanine, which they found first, has 2, UU and UUC.
And is there a pattern to the redundancy?
Often, codons that specify the same amino acid differ only in their 3rd nucleotide. The first 2 letters are often the most important for determining the amino acid. This provides some robustness against mutations.
That's clever. And maybe the most amazing thing about this code.
Its universality. It's nearly universal. With very minor exceptions, the same codons specify the same amino acids in almost all organisms studied, from bacteria to archaea to plants to us.
Wow, from a bacterium to an oak tree to a human, UU means phenylalanine.
Pretty much, yeah. It's incredibly strong evidence for a common ancestor for all life on Earth, and is what makes genetic engineering possible. You can take a gene from one species and put it in another, and the code can still be read.
Okay, let's move to protein synthesis itself. Translation in action, putting the code to work.
Right. This happens on the ribosomes, out in the cytosol mostly, and as you mentioned, it takes a lot of energy.
Before we get to translation, just a quick recap on making the mRNA transcription. RNA polymerase binds where?
It binds to specific DNA sequences upstream of the gene, called promoters. The promoter basically tells the polymerase where to start transcription and which of the two DNA strands to use as the template.
Okay, promoter signals the start, then polymerase moves along the DNA.
Synthesizing an mRNA molecule that's complementary to the DNA template strand. Remember, A pairs with U, G pairs with C.
So if the DNA template strand reads, say, 32ACGTV5,
Then the mRNA transcript will be synthesized as 5AUGCCA3.
right, and the other DNA strand, the non-template one, would be 5'EGCCCA3. So the mRNA looks just like the non-template strand, but with U instead of T.
Exactly. That non-template strand is often called the coding strand for that reason. Transcription stops when the polymerase hits a specific terminator sequence in the DNA.
Okay, so mRNA is made. Now let's look at those tRNA adapters again. The dictionary.
Yes, transfer RNA. These are relatively small RNA molecules, maybe 80 nucleotides long. They fold up into a specific kind of L-shaped or cloverleaf structure in 2D.
And they have two key parts.
Two crucial sites. One is the anticodon. That's a sequence of 3 nucleotides that is complementary to a specific mRNA codon. It's what reads the message.
Anticodon matches the codon.
The other key site is at the 3' end of the tRNA molecule. This is where a specific amino acid attaches.
And how does the right amino acid get attached to the right tRNA?
That's the job of a set of highly specific enzymes called aminoacyl tRNA synthetases. There's essentially one synthetase for each amino acid. It recognizes both the amino acid and the correct tRNA S for that amino acid and links them together. This charting step requires energy, usually from ATP.
So these synthetases are the real translators, in a sense, matching amino acid to anticodon indirectly.
You could definitely argue that, yes.
Yeah.
Crucial for fidelity.
Okay, we have mRNA with codons, charged tRNAs with anticodons and amino acids. Now, the ribosome.
The ribosome, the
Yeah.
workbench, a large complex made of ribosomal RNA, rRNA, and proteins. It has 2 main subunits, a large one and a small one.
And they come together on the mRNA.
Yes. The small subunit has a binding site for the mRNA. The large subunit has binding sites for the tRNAs.
Binding sites, plural.
3 key sites,
Yeah.
typically called the A, P, and E sites. The A site, aminoacyl site, is where the incoming tRNA carrying the next amino acid binds. The P site, peptidyl site, holds the tRNA carrying the growing polypeptide chain. And the E site, exit site, is where the tRNA, having delivered its amino acid, leaves the ribosome.
APE, entry, processing, exit, kind of.
That's a good way to remember it. Now, the actual process of translation occurs in 3 stages: initiation, elongation, and termination.
Okay, stage 1, initiation, getting started.
Right. The small ribosomal subunit binds to the mRNA, typically near the 5' end, and scans along until it finds the start codon. This is almost always AUG.
AUG, that codes for methionine, right?
It does. So the special initiator, tRNA, carrying methionine, or a modified form, FMET, in prokaryotes, binds to the AUG codon. This sets the reading frame for the rest of the message. Then, the large ribosomal subunit joins the complex, positioning the initiator tRNA in the P site. The A site is now open, ready for the next tRNA. This whole setup requires energy, often from GTP.
Okay, initiated. Ribosome assembled on mRNA at the start codon. Initiator tRNA in the P site. Stage 2, elongation, adding amino acids.
Exactly. Now the cycle begins. The ribosome reads the next codon exposed in the A site. The tRNA with a complementary anticodon carrying its specific amino acid binds to the A site.
Matching codon to anticodon again.
Yes. Then the magic happens. A peptide bond is formed between the amino acid on the tRNA in the A site and the growing polypeptide chain attached to the tRNA in the P site. The chain is transferred from the P site tRNA to the A site 2.
So the protein chain grows longer by 1 amino acid.
Correct. Then the ribosome translocates it, shifts 1 codon down the mRNA. This moves the tRNA that was in the P site, now empty, to the E site, where it exits. And it moves the tRNA that was in the A site, now holding the growing chain, into the P site.
Leaving the A site empty again, ready for the next incoming tRNA.
Precisely. And this elongation cycle, codon recognition, peptide bond formation, translocation repeats over and over, adding amino acids one by one as the ribosome moves along the mRNA from 5' to 3'.
Like a little machine reading a tape. Until
the ribosome encounters one of those 3 stop codons, UAG, UAA, or UGA, in the A site.
the ones that don't have matching tRNAs.
Right. There are no tRNAs with anticodons for the stop codons. Instead, proteins called release factors recognize the stop codon in the A site.
Okay, release factors bind, then what?
Binding of the release factor causes the ribosome to add a water molecule instead of an amino acid to the polypeptide chain. This hydrolyzes the bond, releasing the completed polypeptide from the tRNA in the P site.
The protein is free.
Yes. And then the whole complex dissociates the last tRNA leaves, the release factor departs, and the ribosomal subunits separate from the mRNA, ready to start again on another message. That's termination.
Initiation, elongation, termination? Wow. And you mentioned earlier RNA might be catalytic.
Yes. It's now known that the formation of the peptide bond itself, that crucial step during elongation, is actually catalyzed by the ribosomal RNA in the large subunit, not by a ribosomal protein.
So the ribosome is actually a ribozyme, an RNA enzyme.
Largely, yes. This is thought to be strong evidence for an ancient RNA world, predating DNA and complex proteins, where RNA handled both information storage and catalysis.
Fascinating. A glimpse into early life.
Absolutely. Now, connecting this to the bigger picture in eukaryotes, once these polypeptides are made in the cytosol, they often need to get to specific places, right? The nucleus, mitochondria, chloroplasts, ER.
Right, they don't all just stay floating in the cytosol, polypeptide targeting and sorting.
Exactly. There are sophisticated cellular address labels and delivery systems, two main pathways. One is co-translational import. For proteins destined for the endomembrane system, the ER, Golgi lysosomes, vacuoles, plasma membrane translation, actually starts in the cytosol, but then the whole ribosome mRNA complex gets targeted to the surface of the endoplasmic reticulum.
So the protein gets threaded into the ER as it's being made.
Pretty much, yeah. It enters the ER lumen, or it gets embedded in the ER membrane. From the ER, it can then be further processed and shipped via vesicles to other destinations in that system.
Okay, co-translational for the ER and related stuff. What's the other pathway?
Post-translational import. This is for proteins that function in the cytosol itself or need to get into the nucleus, mitochondria, chloroplasts in plants, or peroxisomes.
So these are made completely first.
Yes. Translation finishes on free ribosomes in the cytosol. Then the completed polypeptide, using specific targeting signals within its sequence, is recognized and imported into the correct organelle.
Right. So we know how proteins are made. But like we said, not every gene is turned on all the time in every cell. That's gene regulation.
Exactly. Differential gene expression. It's why a liver cell is different from a brain cell, or in plants, a root cell from a leaf cell, even though they mostly have the same genes.
And this is especially complex in multicellular eukaryotes.
Incredibly complex. And a key piece of evidence for this regulation, rather than just having different genes, comes from plants especially, the phenomenon of totipancy. You can often take a single differentiated plant cell, say from a carrot root, put it in the right conditions, and it can de-differentiate and grow into a whole new carrot plant.
Wow. So that single cell still contained all the genetic information needed for every part of the plant.
Precisely. It means the differences between cell types aren't usually due to missing genes, but due to which genes are switched on, expressed, and which are switched off, silenced. That's gene regulation.
How does the cell control these switches?
There are many levels of control, but a really important one in eukaryotes involves how the DNA itself is packaged. Chromatin condensation.
Chromatin, that's the DNA wrapped around proteins, right?
Yes. DNA wrapped around histone proteins, forming structures called nucleosomes, like beads on a string. This chromatin can exist in different states of compaction. There's euchromatin, which is relatively loose, less condensed. It stains weakly in microscope images. This is where most transcription happens during interphase.
Okay. Euchromatin is open for business.
Generally, yes. Then there's heterochromatin, which stays highly condensed throughout the cell cycle, stains darkly, and is largely transcriptionally inactive. Silenced genes are often found here.
So the basic packaging itself is a form of gene control. Tight packing silences genes.
It's a major factor. The default state for many eukaryotic genes might actually be off due to this packaging. To turn a gene on, you often need to actively remodel the chromatin, loosen it up to make the DNA accessible to RNA polymerase and other transcription factors.
How does the cell loosen it up?
One key mechanism is histone acetylation. Histones have these tails that stick out, which are rich in positively charged amino acids like lysine. Enzymes called acetyltransferases can add acetyl groups to these lysines. Acetyl groups neutralize the positive charge.
Ah, so the histone tails don't grip the negatively charged DNA as tightly.
Exactly. It loosens the chromatin structure, making the DNA more accessible for transcription. Conversely, other enzymes called deacetylases remove acetyl groups. which tends to restore repression.
So acetylation generally means on, decetylation off?
As a general rule, yeah. Another really important modification is DNA methylation.
Adding methyl groups directly to the DNA.
Yes, specifically to cytosine bases, often when they're next to a guanine CpG sequences. Heavily methylated DNA is usually associated with transcriptional silencing. Unmethylated or less methylated DNA tends to be active.
And this is important in plants.
Very important. In plants like Arabidopsis, DNA methylation plays a key role in long-term gene silencing during development, locking cells into specific fates.
Now, these changes, histone acetylation, DNA methylation, they don't change the actual ATGC sequence, do they?
No, they don't change the underlying DNA sequence at all. They're modifications on top of the sequence. And what's fascinating is that these patterns of modification can sometimes be inherited when cells divide. The daughter cells remember whether a gene was supposed to be on or off.
That sounds like inheritance beyond the genes themselves.
It is. This is called epigenetic inheritance. Changes in gene expression potential that are heritable but don't involve changes to the DNA sequence itself. It adds a whole other layer to heredity.
Epigenetics. Wow. So chromatin changes are one big level of control. Anything else?
Oh, yes. Even if the chromatin is open, specific transcription factors proteins need to bind to specific DNA sequences near the gene, like enhancers or silencers, to either promote or block the binding of RNA polymerase. It's a very intricate system of specific protein-DNA interactions.
Okay, let's switch gears slightly to the DNA of the eukaryotic chromosome itself. You mentioned some surprising things earlier.
Right. Two things really stood out to early researchers. First, the sheer amount of DNA per cell, the genome size, varies wildly between species, and it doesn't always correlate neatly with organism complexity.
You gave the Paris japonica example with a huge genome versus tiny plants with tiny genomes.
Exactly. And second, within any given eukaryotic genome, there seems to be a vast excess of DNA.
Meaning most of it doesn't actually code for proteins.
Right. In humans, it might be as little as 1–2%.
Mm-hmm.
Even in plants, it's often less than 10%. This contrasts sharply with bacteria, where almost all the DNA is coding sequence.
So what is all that non-coding DNA doing? Is it just junk?
We used to think maybe, but now we know much of it is functional, even if it doesn't code for protein. A lot of it consists of repeated nucleotide sequences.
Repeats, like the same sequence over and over.
Yeah. Yes, in various forms. You have tandemly repeated DNA, where sequences are arranged head to tail, one after another, like beads on a string. A subset of that is simple sequence repeated DNA, very short repeats, maybe fewer than 10 base pairs long, repeated thousands or millions of times. These are often found in structurally important regions of the chromosome.
Like where?
At the centromeres, which are crucial for chromosome segregation during cell division, and at the telomeres.
Telomeres, those are the protective caps at the ends of chromosomes, right? Like the plastic tips on shoelaces.
Excellent analogy. Yes, they protect the ends from degradation and prevent chromosomes from fusing together. In plants, a common telomere repeat is TTTDG, repeated many times.
Okay, so tandem repeats have structural roles. What other kinds of repeats are there?
Interspersed repeated DNA. These are repeated units, but they're scattered throughout the genome, not clustered together in long arrays.
Scattered repeats, what are they?
The most abundant type are transposable elements, also known as transposons or jumping genes.
Jumping genes. They can move around.
Yes, or make copies of themselves that insert elsewhere in the genome. They've been called molecular parasites because they can replicate and spread within the genome, sometimes disrupting host genes if they land in the wrong place.
That sounds potentially bad.
It can be. But eukaryotes have evolved sophisticated epigenetic mechanisms like DNA methylation to keep most transposons silenced and inactive. However, over evolutionary time, transposons are now thought to have played a major role in shaping genomes and driving evolution, perhaps by shuffling bits of DNA around or creating new regulatory sequences.
So maybe not just parasites, but drivers of change, too.
It's a complex relationship. Now, maybe the biggest surprise regarding eukaryotic genes themselves
Go on.
was the discovery that the protein coding sequences, the actual structural genes, are usually not continuous stretches of DNA. They are interrupted.
Interrupted by what?
By non-coding sequences called introns, intervening sequences.
So the gene is broken up into pieces.
Exactly. The actual coding segments, the parts that eventually get translated into protein, are called exons, express sequences. Exons are separated by introns.
Exons code, introns interrupt. When was this discovered?
Around 1977, it was a huge shock. Scientists were comparing mRNA molecules to the DNA genes that coded for them. They expected them to match up perfectly. But when they hybridized mRNA to the DNA gene and looked under an electron microscope, they saw loops of DNA sticking out.
Loops.
Yes. The mRNA only bound to the exon regions of the DNA. The intron regions of the DNA looped out because they had no complementary sequence in the mature mRNA. That visually proved introns existed and were removed from the RNA.
Wow. And this is common in eukaryotes.
Very common in multicellular eukaryotes. Most protein-coding genes have introns, sometimes many of them, and they can be much longer than the exons. Introns are also found in some tRNA and RNA genes. Prokaryotes, however, rarely have introns.
Why have introns? Do they do anything?
That's still debated. But one popular idea is that having genes split into exons might have accelerated evolution by allowing exon shuffling.
Shuffling exons, like mixing and matching protein domains.
Exactly. Recombining existing exons in new ways could create novel proteins with new functions much faster than waiting for random mutations to achieve the same thing.
Okay, so genes have introns that need to be removed from the RNA. This leads us to transcription and processing of mRNA in eukaryotes. It's more than just making the copy.
Definitely more complicated than in prokaryotes. First, as we touched on, transcription and translation are separated in space and time. In eukaryotes, transcription in the nucleus, translation in the cytoplasm.
Unlike prokaryotes, where it can happen simultaneously.
Right. Also, in eukaryotes, each structural gene is usually transcribed individually with its own promoter and regulatory elements. Prokaryotes often group related genes together into operons, transcribed as a single unit.
Okay, separate transcription. What happens to the RNA transcript after it's made by RNA polymerase in the nucleus?
It's not ready for translation yet. It's actually called pre-mRNA at this stage. It has to undergo several processing steps before it can be exported from the nucleus as mature mRNA.
Processing, like what?
Three main things. First, a special modified nucleotide, a cap, is added to the 5' end of the pre-mRNA. This 5-foot cap is crucial later for ribosome binding and protects the mRNA from degradation.
Okay, a cap on the front end.
Second, at the 3' end, an enzyme adds a long string of adenine nucleotides, typically 50 to 250 of them. This is called the poly-A tail.
Poly-A tail on the back end, what does that do?
It also helps protect the mRNA from degradation, aids in its export from the nucleus, and plays a role in initiating translation.
Cap and tail for protection and function. What's the third, maybe most dramatic step?
Splicing. This is where those non-coding introns are removed, and the exons are precisely joined or spliced together.
Cutting out the introns and stitching the exons together.
Exactly. This has to be incredibly precise. If the splicing is off by even a single nucleotide, it will shift the reading frame for all subsequent codons,
leading to a completely different and likely useless protein. Garbled message.
Right. This splicing is carried out by a large molecular machine called the splicem, which is composed of small nuclear RNAs, siRNAs, and proteins. Again, RNA playing a catalytic role here too.
So pre-mRNA gets capped, tailed, and spliced to become mature mRNA ready for export and translation.
Correct. But there's another layer of complexity here too.
Oh.
Alternative splicing. Sometimes the same pre-mRNA transcript can be spliced in different ways. Meaning that what counts as an exon in one version might be treated as part of an intron and skipped in another version. Or an intron might sometimes be included.
So you can get different mature mRNAs and therefore different proteins from the same initial gene transcript.
Exactly. Alternative splicing vastly increases the protein diversity an organism can generate from a limited number of genes. It really blurs the lines of what we even define as a gene or an intron. One gene can code for multiple related proteins.
That's amazing flexibility.
It is. And it adds to this picture that the eukaryotic genome isn't static. We talked about transposons moving around. Viruses can insert DNA. Bacteria can pick up DNA from the environment. The idea of a perfectly stable chromosome is, well, not entirely accurate. DNA can be quite dynamic.
Okay, one last piece of the puzzle you mentioned, non-coding RNAs and gene regulation. Beyond mRNA, tRNA, rRNA.
Yes, this is a relatively newer area of discovery, but incredibly important. We now know there's a whole world of RNA molecules that don't code for protein, but play crucial regulatory roles.
Non-coding RNAs regulating genes. How?
One major class is microRNAs, or miRNAs. These are very small RNA molecules, typically about 22 nucleotides long.
Tiny RNAs, what do they do?
They primarily act to decrease gene expression, often by binding to complementary sequences in specific messenger RNAs. This process is called RNA interference, RNAi.
So the miRNA finds a target mRNA.
Yes, and that binding can either block the translation of the mRNA into protein or can lead to the mRNA being targeted for degradation, chopped up. Either way, less protein is made from that gene.
So they act like fine-tuning knobs, dialing down gene expression.
That's a great way to think about it. And they're involved in regulating huge numbers of genes and critical for normal development. If mirin regulation goes wrong, you can see major problems.
Like the leaf curling example in Arabidopsis.
Exactly. A specific mirin normally regulates a gene involved in leaf development. If that mirin isn't expressed properly, the target gene is overactive, leading to abnormal curled leaves.
Wow. Are there other types of regulatory non-coding RNAs?
Yes, other classes exist, like small interfering RNAs, siRNAs, which are also involved in RNA, often as a defense against viruses or transposons. And there are longer non-coding RNAs that can influence chromatin structure, helping to establish or maintain silence regions like heterochromatin.
So RNA isn't just a messenger or adapter, it's a major player in regulating the genome itself.
Absolutely. The regulatory web is incredibly complex, involving interactions between DNA, proteins, and a whole zoo of different RNA molecules.
Wow. Okay, what an incredible deep dive that was. We've gone from the double helix
Its elegant structure and replication,
through transcription and the complexities of translation,
the genetic code, the ribosome's workings,
to the layers upon layers of gene regulation in eukaryotes, chromatin, epigenetics, transcription factors, alternative splicing.
and finally these non-coding RNAs adding yet another layer of control.
It really underscores the incredible complexity, but also the elegance of how life stores, copies, and expresses its information.
It does. From the simplest base pair rule, A with T, G with C, emerges all this intricate machinery and regulation.
Makes you really appreciate how these tiny molecular events scale up to create the specificity of a leaf cell, a root cell, the whole diversity of plant life, and really all life.
Absolutely. And thinking about all this, the dynamic nature of DNA with transposons, the flexibility of alternative splicing, the layers of epigenetic control, it does raise interesting questions, doesn't it?
Like what?
Well, given this inherent flexibility and dynamism in how genetic information is used and even organized, what does that imply about evolution? Does this built-in potential for variation and regulation allow species to adapt more readily to changing conditions?
That's a fantastic thought to leave our listeners with. How does this molecular toolkit translate into evolutionary potential? So much to ponder. We really hope this deep dive has given you, our listener, a clearer and more engaging understanding of these absolutely vital biological processes. Thank you so much for joining us on this exploration. We really appreciate you spending your time with the deep dive.